is a very specific member of the Mycobacterium avium complex. The total genome sequence determined in the Genomics Center at the University of Minnesota shows a circular DNA molecule, here shown in green, with 4.8 million GC-rich base pairs, predicting 4,350 open reading frames. Throughout the genome, mostly in these green areas, there are up to about 47 insertion elements throughout the genome, of which the most frequent in 14 to 18 copies is one called IS900 that is inserted in highly conserved sites throughout the genome, and in all but one case enters the genome between the ribosomal binding site and the start codon of the target gene. And since it possesses its own promoter, these elements have the ability to manipulate genetic expression in the areas in which they're inserted. You will note that there are some white areas in the genome. The green ones are virtually identical in genetic organization and in DNA sequence to what I shall call garden variety M. avium, that rarely cause disease in animals and humans unless they're immunodeficient or otherwise debilitated. These white areas here consist of genes, sometimes with a reduced GC content suggestive of acquisition by lateral transfer, that encode for pathogenicity determinants in other organisms, or indeed are unique to these pathogens. It is tempting to think, therefore, that the widespread pathogenic phenotype of this particular subspecies of the M. avium resides in these small percentage of the total genome, together with what the insertion elements can do to the expression of the common part. Now, these MAP is a multi-host chronic enteric pathogen, and it can infect and cause disease in a very wide range of species, from ruminants all the way through to primates. The principal reservoir of these organisms in our communities is in our domestic livestock. But wild ruminants, and together they are foregut fermenters, are also involved. Alpaca and horses, which are examples of hindgut fermenters. Monogastric, such as pigs, wild boar, dogs, cats, can all get chronic inflammation of the intestine. Rabbits and their predators, including insects, chickens and carrion birds. I picked a wild one here, and bears. It is a true multi-host chronic enteric and systemic pathogen that can cause systemic infection and initiate and maintain chronic inflammation of the intestine of a range of histopathological types in very many species. It can also exist in animals for years without causing clinical disease. It emerged first in its description in 1895 by Herr Professor Joni in Dresden. And what happened after then is two things. One is the population of Western Europe and the food animals required to feed them increased rapidly. At the same time, descriptions occurred from many countries of the occurrence of Johne's disease, and in the 30s and 40s, the reviews showed that the incidence of clinical disease in dairy herds was in the range 15 to 20%. This high incidence of clinical disease appeared to diminish, and what we have now is a very high level of subclinical infection in our domestic livestock. These figures here for individual countries, naught for Sweden with a question mark, and I don't know the data from France. The figure on the left is the herd prevalence, and the one on the right is the individual animal infection rate. The data are not terribly good, but we do have over time data for Austria and also from the Czech Republic showing that the infection in animals is increasing rapidly. This is a particularly important study in Holland where they took 90 herds that had never had paratuberculosis, never tested for paratuberculosis, and they tested them by fecal culture every 6 months for 4.5 years. And at the end of this time, 61% of the herds were found to be infected. A recent study over the last 3 years performed by the Department of Agriculture in Britain has found a 57% prevalence of infection in our dairy herds. The same situation holds in the North American continent, where the first description was in 1908. There are multiple data from many areas from Canada and the United States, and increasingly from Mexico, showing the herd and single animal prevalence. But these have perhaps been superseded in our understanding of the scale of this problem when we look at the results from the US Department of Agriculture National Animal Health Monitoring System, performed in 2007 and published in April this year. And what they did was to study 524 farms in 17 major dairy-producing states, representing a very wide range of climatic conditions. And on these farms, they took manure samples from 6 common areas, such as pens, alleyways, milking parlour, the slurry pits, and so on. They cultured them on Herald egg yolk media for only 8 weeks, and then did PCR for paratuberculosis on the alleyways. And the overall 68.1% of a sample of dairy herds in the United States, representing 80% of your milk production, tested positive for these organisms. There was a well-known relationship with herd size, such that herds of 500 cattle or more, 95% of them were infected, and on 25% of all farms, they were MAP positive in all 6 areas, indicating a very high level of infection. Of course, we welcome more data, but at the present state of our knowledge, it is, if you like, a done deal that MAP infection is widespread in the domestic livestock of Europe and North America. It's also prevalent in those countries which these northern continents traded, particularly in the 19th and 20th centuries, such as Argentina and Brazil, South Africa, Australia, New Zealand. But now Johne's disease and MAP infection is spreading to former low incidence areas. It's spread, for example, following the fall of the Iron Curtain into Eastern Europe. It's occurred in Egypt, in Iran, in India, particularly in goats, in Korea, in Japan, and in China, which has been importing huge numbers of animals from Canada. Johne's disease and MAP infection, therefore, is a new disease, recognized first at the junction of the 19th and 20th centuries, and is now expanding globally. This is an animal with Johne's disease in a herd of identical animals. These animals are almost certainly subclinically infected. This animal is shedding gazillions of these organisms onto pastures, which infect rabbits, travel up the rabbit predator chain. These grazing animals eat the rabbit pellets and become infected in the cycle of environmental contamination. If you look at the intestine of this animal, it's very obviously filled with macrophages stuffed with typical classical zeal bacteria, in this case, MAP. This animal also has Johne's disease, and its gut looks like this, in which you can't see any mycobacteria, or you see Zn positive mycobacteria, staining mycobacteria lineages, including these classicals of inflammatory. The cell classical repertoire of C is the positives. So one of the first things we learn about this organism and its variability is the fact that it can cause a range of histopathological diseases in animals, very much like the range between the lepromatous and the tuberculoid forms of leprosy in humans. Now, there's another thing about this nature of this organism that I'd like to draw to your attention. And this is the unreliability of culture as a general diagnostic. And this is the photograph of a 6-month-old culture of an infected cow near Carmarthen in Wales. You see the organism growing in its characteristic clumps. And microscopically, you see the clumps of typical Zn positive organisms.